All right, so all we have, let's go back. So we just keep doing this where we're pulling the thick filament, the myosin out of the thick filament is pulling on the actin in the thin filament. We're shortening the sarcomere until our brain or our spinal cord says, all right, that's enough. When the signal goes away, our brain and our spinal cord stop the signal. What happens is the action potential ends. And immediately upon when the action potential ends, your muscle cell pumps all that salt or all that sodium out. And as soon as it does that, it overshoots and it goes negative again. And it goes really quite negative. And when that happens, the plug is put back into the calcium store within the cell. And then calcium is actively pumped back out of the muscle into the calcium store site, the storage site. And it's done so by this protein called SERCA. SERCA, cool. All it means is a calcium, Ca. So it's the sarcoplasmic reticulum calcium pump. You don't have to know that. All you have to know is that it's SERCA. And SERCA is just a pump. And just like myosin uses ATP to do something, SERCA uses ATP to pump all of the calcium up into this little storage site within the muscle. And the storage site is there because when the next action potential comes, we'll pull this little plug and we'll open up the storage site and that'll flood out and start the cycle again. All right? So basically, your brain starts a muscle contraction by causing a positive charge or sodium to come in, causing a positive charge, which is going to pull the plug on this little storage site of calcium. Calcium is going to flood into the muscle, bind to troponin C, pull troponins and the tropomyosin out of the way, allow myosin to bind to actin. When it does, there's going to be lots of power strokes. As long as there's ATP around, we're going to pull on this way, we're going to pull on it this way, and all of those things are going to bring the sarcomere shorter or bring the Z lines closer together. And now what we've done is we've caused muscle contraction. And then when we do that in each sarcomere throughout the muscle, now the whole muscle is contracted, or that whole muscle fiber is contracted. And when we do that across different muscle fibers in the fascicle is contracted. When we do that throughout the muscle itself, all of the different fascicles are shortening, the muscle will shorten itself. All right? So what I told you in these lectures is that there's three types of muscle. We have skeletal, cardiac, and smooth muscle. Two of them are striated, one is smooth. Two of them are involuntary, one is voluntary. And two of them have only one muscle or one cell to make up the tissue to eat the contractile unit within the tissue. And the other one has a bunch of cells that have come together and fused. All right? Cardiac and skeletal are striated. Cardiac and smooth are involuntary. Skeletal muscle I showed you is made up of fibers. Those fibers are surrounded by perimysium. Endomysium. The fascicles are groups of fibers that are surrounded by perimysium. The fascicles are brought together into muscles, which are surrounded by epimysium. Okay, the thin filament has actin in it, which has the myosin binding site. It has troponin. It has tropomyosin in it, and it has the troponins C, T, and I. When calcium enters the muscle, it's going to bind to the troponin C, and troponin C is going to change its shape. And when it changes its shape, it pulls its binding partners or its friends, tropomyosin and the other troponins, pulls it out of the way. And now the myosin binding site on actin, it becomes revealed. And as soon as it's revealed, now myosin can bind to actin. And when it does, it goes through what's called the power stroke. It binds to actin, it goes through the power stroke, and when it's in the power stroke, it kicks off that low energy molecule ADP. And it only can release actin when it binds to that high energy molecule ATP. And when it binds to ATP, it releases the actin, and then it's going to break the ATP molecule down to allow it to recock or to re-extend itself. And now when that happens, now it's ready to go again. And if calcium's still there, The binding site is still open, so now it's going to bind again, power stroke, release ADP, get new ATP, release the actin, break it, come back up. And it's going to continue to do this power stroke until you remove the calcium, and the calcium is taken up by this protein called SERCA, and it's then put back into this storage area for the next time your brain or your spinal cord wants to activate that muscle fiber. All right? I know there's a lot of complex stuff in there, especially for people who don't do science so much, but hopefully, if you go back through it and if you have any questions, please let me know. But hopefully, this is a little bit clearer as to how a muscle works at a cellular level. All right?